for instance, so we've talked about, windows like a 12 a Thomson window, cycle time, which would be, we talked about the time needed to run through all of those windows and then start back again. Whether you do gas-phase fractions or not to make a library, and Mike gave a good pitch of why he does that. And then we've talked about the speed and the sensitivity of the instrument, which is kind of, it's they're kind of one and the same thing. If your instrument's more sensitive, then you can run it faster because it doesn't take as many ions. But the big key, I think, is again, the chromatography, is matching all of that back to your chromatography to make sure that you get this, what we call in points across the peak. How many times the transitions or fragments are measured so that we can get a good a good area. So I made this next couple of slides here just as a recap here, but you got this trade-off of how you do this, and I think that's the fun part of being a scientist, is you're in the lab and you're trying different things, and unfortunately here at the National Resource, sometimes we find something that works and then we run with that, and it's hard sometimes to do the development side when you're running a lot of samples, but that's why this has been so good for us to talk about all these different things. I haven't patented this yet, so if you want to steal this, I would be so flattered, but I made this up. I thought, well, why is it so important that we're so interested in the points across the peak, trying to find that at the very bottom of this triangle, the area of the fragment ions? How important is that? Because that really is what we're using to calculate the intensity of the peptides, which is then used to figure out the abundance of the proteins, which is then used in the differential expression within the groups and between the groups, which is then used by somebody to do biological conclusions for the experiment of what real reality is. And then that is actually directing research and treatment. And so it's a lot. I mean, it's very, very important that we get this right. So one of the questions is basically, how do I validate a protein that has been shown to be differentially expressed in a volcano plot? So I'm just going to kind of throw some of this here and kind of walk you through this. This is the volcano plot that you see, and the plot over on the right is a part of the software that Stephanie Byram and her team here at the National Resource put together as part of our bioinformatics workflow. But a lot of times, this plot, that volcano plot, is the most important thing that the PI is interested in. And they immediately, their eyes zoom in over into the red or the blue dots, each one representing the different proteins, and they go, ooh, and they click on it. We have an interface where you can click on it, and you can see this differential analysis over here on the side, and it'll bring up some of the information there, the log fold change and that. Well, how do you validate that? Well, the old school way in the Qual Browser would be to have to actually go in and interrogate the data, to find a certain peptide. So I'm just going to do this real quick, and then we're going to take a break here. But for instance, if you have a particular protein that you found here, and the one we're going to chase down is this called serapine B9, and it's found in our JGN3 lysate. That's a myeloma cell line that one of the researchers on our floor grows that up for us, and we're able to use as our standard. And so we this particular experiment had a staggered window approach. So you can see the windows of those little gray platforms moving down, and they're offset by about 6 Thomsons there. So if I open up my Qual Browser there and I look down deep there, I can find the MS signal for this particular peptide, which is in that protein. And you can go in and you can figure out what the charge is on that guy by looking at the isotopic numbers up there. And if you scan across there, you can see essentially how many points that you would have, how many cycle times. So each, for this particular example, each one of those is an MS scan. And so we do if we go back to here, we do one MS scan, and then we do all those DIA windows. Then we do one MS scan, and then we do all those DIA windows. So I'm pretty confident that if I have 1, 2, 3, 4, 5 of those MS scans, then I'm going to also have an analogous MS2 scan in between there that would cover that particular peptide. And maybe this would be a conversation for, tomorrow or something, but it seems like we go to a lot of trouble to not quantitate off of the MS scan. And we go to a lot of trouble to try to quantitate off the MS2, which has its own problems, as we've talked about. So, so trying to find the particular precursor has we're looking for a window that has the mass of that precursor in it. And so, if I could just quantitate on that, I could just draw a little triangle like that. The other, so if I find and I go through and I look at the actual MS2 windows, we've seen this before, but this is a pretty complex window that has a lot of fragments in there. So there were lots of precursors that were fragmented during this one time window. And because I know the answer, I can go back and say, well, I know that really tall guy right there at 1256 happens to be the Y10 ion of the guy that I'm looking for. So if we actually look for that, we can then find these in the first. So I have 2 DIA experiments going on that are staggered. And so essentially I'm picking up about 4 or 3, if you count like Rick does, 3 signals in the first DIA experiment, and I only get 3 in the bottom. And so together I get about 6 across the peak there. And that I think, I don't know how many years ago, but that's how you did it, right? That's how you would figure out that. Well, so a couple of things that we can point out. This little picture here on the bottom, going across the bottom, that is from the output from Spectronaut. So if I just typed in what the protein label was for that, it will pull that up and it shows me all the different experiments that I had. I had some staggered and some non-staggered. And I haven't talked about this, but I just took 2 different cell lines and I compared them to each other, HeLa and this JJN3, so that I would get lots of differential expressed proteins in an easy way. What I didn't realize, because I'm not the biostats person, but I have to give a shout out, is that every time I did this, it would yell at me because it says, hey, you have too many to fit the model, which is I think it's like 20% or something, so the idea is that you're not gonna have your proteomic changes aren't going to be dramatic in more than 20% of your proteins expressed there. So this particular guy here, if you look, you can see that we only got this thing measured one time in the no staggered or in the JJN3. I'm sorry, in the HeLa experiment, we only got it measured once, but we got it measured a bunch of times in the JJN3 because it's much more abundant there. So but this picture over here is what a lot of people base their biology on, is just looking at, I've got these little red dots up here that all are grouped together. So I've got good within the group or small variation within the group, but then I've got a large distance between the 2 groups, and that's great. But there's more information there that we need to be able to make that decision. Now, if we go in deeper in Spectronaut, it'll show us, and lots of the other things will as well. It shows that there are actually 22 precursors that were measured for this particular protein. So, why are we having this conversation, right? Obviously it's there. There's plenty of things to quantitate on, and that one particular peptide is just one of many things that we could focus in on. Well, here's the other interesting thing here is if we go back and we look at some of these other transitions, so from the original MS2 in that window, you can see that there are three easily identified fragments from this particular peptide that if you, the picture over there on the left that has the MS and the MS2 fragments, again, that's a picture from Spectronaut. And you can see that those line up. And if you actually want to, you can go and you can see about 6 points across each peak there. And that's what we're getting our quantitation of. So, this whole story is to say, that for me to validate one particular protein or one peptide from one protein and one experiment, then something like this needs to be done for me to believe that this is a truth. And I guess the question is what, I mean, we kind of know Mike's end, I think he kind of feels the same way, that the interrogating the data has to be done. But I guess the question is who has time to do all of that, right? Because I think as a core people, right, you're presenting the data to the biologist saying, there's your data. And they're saying, yeah, but what does it mean? And is this a good point here? Is that a great protein, based upon that? So
also because it's an interactive process. They look at those, they know their biology, and they say, this is an exciting result, and would ideally come back to you and say, I just want to double check. Mm-hmm. How did you come to this? And maybe even ask for a chromatic range.
That's a great point. Which he's gonna Steve's gonna send everybody. Mike likes to interrogate the data. Yeah. But I mean, that goes back to the original thing that we started, like said, one of the first things we said this morning was about communicating all of these objectives and stuff, because you don't want to have to do the experiment twice, right? You don't want to take the, oops, you really, can't run statistics with an N of 2. So communicating that on the front end, obviously. I don't want to put anybody on the spot, but how much interrogating are we doing of the data that we're pushing out? Is that tend to fall on you guys and what you do, or is that something kind of like Mike said, you kind of give it to them and you kind of hope they come back with questions, and then you're more willing to have that conversation with them then? They give it to me. Oh, that's why they hire a bioinformatics guy, right?
Yeah, I think, well, first of all, I have to disagree with your very first statement where you said that you guys managed to do more development and run a high throughput of samples than any other facility I know. So you should not downplay that, because I mean, I know from being on the review committee how much samples come through here, and you guys still do an amazing job of evaluating things and developing things. So I think that that's awesome. And I think that's necessary to be the successful core. You have to find that balance, right? You can't just go with all the published stuff. You lose out, right? I mean, and so I think you and your team and, and Rick and you guys have done an amazing job.
Andy, can we get that in writing?
Yes.
Yeah. Yes. Can you speak into the microphone?
But yeah, I mean, in terms of we're in the same boat, right? Because there's just not enough time to really go through every piece of data the way you would want to, right? So you start to set these thresholds and these ideas, and a lot of it comes down to, we have these long discussions about, especially with the DIA data, how we're going to present it to the, investigator, and what sort of caveats are we going to have before they, because it's like, we believe this is changing, but before you go making a knockout animal of this model or spending all this time developing an antibody, come back to us, and then we will do it. I mean, we can't do it for every single one, right? But we can set some sort of thresholds where if you believe this is interesting, you want to pursue it further, come back to us. We can dig a little deeper into the data, and I think it has to be this iterative process back and forth. You want to share things, but then you want them to come back to you and say, yeah, this is interesting. And then we can dig deeper, because as the data gets more, like I said, now, I mean, it's bad enough as it is right now. You're going to buy National, and now you're going to run, 60 samples a day. I mean, we're barely surviving off 10 samples a day with manually looking at the data. So as that grows, there has to be some sort of tiered system. I think that and this could be a whole nother meeting where we all think about moving forward, how to present the initial data with what qualifications to what you should do with that before coming back to us. Because I mean, it's only going to get worse as the instruments get faster and the turnaround, we get more and more data. I mean, it has to become more and more automated, and we have to start to release some of our, well, at least mine, some of our deep-seated, beliefs that you actually still need to look at a raw file before you send a report. But at some point, that's, there's going to be some, yeah.
Well, and that,
Yeah.
and that creeps into our conversations here, where what we've done, and we'll talk about this more tomorrow when we talk about managing, and one of the best things I think we're doing is we are pushing the consults and the sample lists and the what contrasts that do you want to, you're not just sending us the samples and then say, yeah, and when I see the data, we'll figure out what we're doing, after you run it. That doesn't, nope. No, because there's too much at stake.
Yeah.
And the other comment is that the re-education of people that this is a collaborative field. You don't do proteomics in a vacuum, right? That they say, oh, I'll just send my samples to you and I'll get the data back.
One man just identify it, right? Those days are gone, right?
I have one question. What tips or advice, because you guys have a huge technology, but sometimes I struggle with people who really don't trust. They say, I want to do this analysis, but Radio Hall or something told me that the best way to do is this. When you know that sometimes you can do label free without spending too much money, you can do it, but they trust more in the Radio Hall or the paper or whatever say. What do you think? Is it time to try to cross with somebody else to say, can you support to me to convince this guy that really what we are offering is okay, at least at the beginning? Because this is another point before to generate any other thing, to provide the service. Because they say, no, somebody told me that label free is junk. And how you convince this guy to avoid kick him from the facility? Because somebody will say, no, that's it, leave.
Yeah, no, I think that's true. We still, I mean, we have that all the time, so I think some ways to handle that are to refer them to, we send them directly papers using the methodology that we want to use, whether if we're on the paper, that's even better. But if not, there are other papers out there to do that. And, but there's always going to be, I mean, we always come across a couple of people that want to work with us that are just set in their ways, right? They just determined that, the way so-and-so does it is the way they want to do it. And, if we're able to do it, most of the time we'll do it, but a lot of times we're able to convince them just with a little bit of, sending them some paper, say, yeah, that is a way to do it. But, in our center, we've been much more successful this way. And we could try it that way, but, just sort of trying to sway them to say like, well, our center's set up better to be successful doing it this way. We kind of could do it this way, but that's not what we're strong at. So you probably want to, play to our strengths and we'll do it that way. But yeah, I mean, it is tough. I mean, some people just are really stuck in their ways.
Yeah, I had one experience the last time that the guy was so annoying because he said, I wanted to do this. Don't complicate your life. Go and do a Western blot. Because really, you're looking for that kind of thing, don't you?
Yeah.
And they say, why? Because you have proteome. You are threw away your business. No, I am trying to be sensitive. Don't make complicated your life. If you are looking for that particular protein, you have the antibody. Do a Western blot. That's it.
Yeah, I mean, ultimately, it's the researcher's decision on what, how to do the experiment. And so you can advise all you want. But I mean, it's their, you can't make them, do something. So, we have people that say, okay, we'd get better data doing DIA for this. Like, we want to do DDA because we're comfortable with it. We want to run a gel and do DDA because we've done it for 10 years and we like the data and we understand the data, knowing that it's going to be better if we do DIA, but we had planned on shutting down an instrument that was only doing GELC, 3 or 4 years ago, and it stays busy because of the legacy people that don't want to change. So,
Sometimes, you know, what I found is he say, give me a possibility to do one try and I will show you. And at the end of the day, they come and say, sorry, I apologize what I say, because look at that, you can get this. But sometimes you don't have that option because you are, for example, in my case, I know that I cannot handle level 3 with 16 group. You know, and that will say, no, go and I can make a bridge with a guy here or wherever. They can do TMT for you. And I can work with you at the beginning and at the end to figure out how to do it. But sometimes it's hard because they put it to you in a situation to say, you don't know nothing about it. Why? No, I can tell you how you say, it's my experience can work, you know.
yeah.